Hello, this is our third video lesson on mass spectrometry. In our first video lesson, we talked about mass spectrometry in general, and we talked about different ionization methods. And in the second video lesson, we talked to you about mass analysis, about types of mass analysis, 6 different types of mass analysis. And in this video lesson, I try to deliver some info about some hybrid types of mass spectrometers that are widely used in application, in practice, and some mass spectral analysis of biomicromolecules, biomolecules, low molecular compounds are discussed at the end of the lecture. Here we go. Okay. In the second lesson, we talked about the mass analysis, and according to mass analysis and tandem classifications, we divided mass spectrometers into GCMS, LCMS, yeah, according to the applications in tandem with gas chromatography or LCMS. And according to instrument design, we said that mass spectrometers can be of single or tandem applications like single quadrupole or triple quadrupole. And according to ionization methods, we said that mass spectrometers could be provided with electron spray ionization or multi-matrix assisted laser desorption ionization, electron ionization, or chemical ionization methods, techniques, and mass analysis, where, as we talked, could be quadrupole, TOF, orbitrap, trap, and others. And now we are going to see more about the hybrid types of mass spectrometers. Okay, the first one is multi-TOF. Okay, this may not be a kind of hybrid because matrix assisted laser desorption ionization is the type of ionization method, and TOF is mass analyzer. So multi-TOF is something like this that we talked in our lesson, that neutral matrix is mixed with the analyte and is subjected to laser treatment. And under the laser treatment, these two types of molecules, analyte and matrix, turn into the gaseous state. They could be ionized or non-ionized. So this is how MALDI works. And TOF is time of flight, is the type of mass analyzer. And MALDI is a soft ionization technique to create ions with minimal fragmentation by using laser energy. In TOF, the protonated ions are accelerated by an electric field to make an ion have the same kinetic energy as any other ions that have the same charge. The velocity of the ion depends on the mass to charge ratio, and the time it takes for the ion to reach a detector is measured. MALDI-TOF is a mass spectrometer that can analyze a wide variety of biomolecules, including peptides and carbohydrates. And in this video lesson, now we can see the work mechanism of the MALDI-TOF. Okay, here in this video, we watched how the MALDI-TOF works and identifies the ions. Okay, next. Oh, I'm sorry. Okay, the next one is triple quadruple mass spectrometry. Here we can see three consecutive quadruples, which uh, causes the name triple. Uh, Triple quadruple, as we talked in the second lesson, it provides us with much more, let's say, features that by using triple quadruple mass spectrometers, we can identify and quantify low abundant compounds as well. So triple quadruple mass spectrometer is a tandem mass spectrometer in which the first and the third quadruples act as mass filters. Here we can see the selection and selection. So it's written here, the first and the third quadruples act as mass filters, and the second act as a collision cell to fragmenters. Here the to fragmenters, collision cell to fragments happens, and collision to fragment the selected precursor parent ions and to generate fragment daughter ions. So by using triple quadruple mass spectrometry, the fragments of daughter ions can be identified and quantified. The triple quadruple mass spectrometer is probably the most famous and widely used tandem mass spectrometer, which is relatively simple and easy to use with good reproducibility to offer various applications at a low cost. The triple quadruple mass spectrometer can be used for structural elucidation, which can provide information about fragmentation patterns as indicated here. And due to increased sensitivity and specificity yielding lower detection and quantification limits, triple quadruple mass spectrometer is a vital option for many areas such as pharmaceutical development, clinical research, environmental studies, and other disciplines of science. So again, triple quadruple mass spectrometer is something that by using of which we can detect and quantify low abundant compounds as well. Okay, here we can see Agilent 64985 C triple quadruple LCMS tool, and we can find out how it works. Okay, this is about, sorry, this video was about Agilent 6495 C triple quadruple LCMS, and the next one is quadruple orbitrap mass spectrometer. Okay, in the quadruple orbitrap mass spectrometer that combines the high-performance quadruple precursor selection with high-resolution, accurate mass orbitrap detection. The first mass analyzer is a quadruple, and the second one is a high-resolution orbitrap. Here we can see here is the quadruple, and this one is orbitrap. This tandem allows more accurate and high resolution results. It can be widely used in various fields like proteomics, metabolomics, food safety, toxicology, and so on. Based on two mass analysis, quadruple and orbitrap, we can claim more efficacy of this kind of tool in proteomics. At creative proteomics, we can provide various services by using quadruple orbitrap mass spectrometry like Q exactive hybrid quadruple orbitrap mass spectrometer and Q exactly plus hybrid quadruple orbitrap mass spectrometer. The quadruple orbitrap mass spectrometer can combine high performance quadruple precursor selection with high resolution and accurate mass orbitrap detection, and the precursor ions can be dissociated with high energy collision induced dissociation. So this is another widely used mass spectrometer. And another one that was provided with tandem is quadruple trap mass spectrometer. Quadruple trap mass spectrometer is a hybrid triple mass spectrometer, different from classical triple quadruple mass spectrometry. The Q3 can work as either a standard quadruple mass filter or a linear ion trap with higher sensitivity than traditional 3D ion trap. This instrument retains the classical triple quadruple scan functions like selective reaction monitoring, and it can also be used for sensitive ion trap experiments. This kind of, let's say, sequence of the elements we can see inside the quadruple trap mass spectrometer. Okay, next. And another one is the hybrid linear ion trap orbitrap mass spectrometer. It's one of the Fourier transform-based hybrid instruments, while the first mass analyzer is linear ion trap, and the second one is a high resolution orbitrap. So here we can see the quadruple part, and this is octuple as we watched in the last video. And here is a high pressure cell, low pressure cell, and here we can see a quadruple mass filter. And after this, again, after triple quadruple, here we can see the orbitrap. Yeah, not triple quadruple. Yeah, I think triple quadruple. The first one, the second one, and the third one. Okay, quadruple. Yeah, maybe double quadruple. And after the quadruple, here we can see the trap, orbit trap, and these two mass analyzers allow this tool to achieve highly, let's say, high resolution results. The linear ion trap uses a set of quadruple rods to confine ions radially and a static electrical potential on the end electrodes to confine the ions actually. The orbit trap consists of an actually symmetrical mass analyzer. This hybrid mass spectrometer is widely used in proteomics and metabolomics analysis. So based on this result, as we have been discussing, that we can make a conclusion that mass spectrometers are especially very useful for proteomics and metabolomics analysis. And this one is the rare one, Fourier transform ion cyclotron resonance. In the second video lesson, we said that this tool is very expensive, and the results, highly resolution results can be obtained by using this tool. Okay, now we are going to see how it works. It was not provided with the sound. Here we can see the process where the electrospray ionization can happen. Here we can see the quadrupole, triple quadrupole process. Here the selection is provided. And inside this orbitrap here, we can see radio frequencies. These radio frequencies can give different oscillations, and based on this kind of results resulting from different molecules, amplitude and frequency based results are obtained and converted into mass to charge results. So based on the oscillation, this kind of amplitude and frequency related data is obtained and converted into mass to charge data. So this is how this kind of tools work and results are obtained. And now in a few pages we are going to see the results obtained from the papers. The first one, these results were taken from the paper named flavonoid profile and antioxidant properties of Algerian common U, Taxus baccata. Here we can see that the structure was established by using LCMS, by using the HPLC. The compounds were separated from each other. Here we can see the belongings of the peaks to different compounds. 14, for example, belongs to quercetin, 3 retinol derivative. 24, for example, belongs to unidentified compound. Okay, not all compounds were identified in this work. And 26, for example, this peak belongs to the cytopitocin. And by using the mass spectral data, for example, when this peak was sent to mass spectral analysis, so this kind of structure could be suggested based on the, let's say, split of this kind of charged molecule into smaller fragments after ionization, let's say. Here we can see that these two molecular ions differ from each other by this kind of group. Okay, the subtraction of this group of atoms lead the formation of this kind of molecular ion from this. So from this ion, okay, we can see that the authors were claiming the formation of these two ions as well. And up to my understanding, here the authors used LC-MS results. So the mass spectral data obtained from this were also subjected to MS analysis. And here we can see the MS data resulting from this kind of molecular ion by subtracting this kind of triple let's say, hydroxy derivative of benzene. So these kind of mechanisms explain the formation of the ions of small molecular ions of small, I mean lower molecular compounds. In high molecular compounds, okay, different approaches are used. Okay, here we can see some results from another paper. And here we can see that these peaks belong to different compounds. The peak 12, for example, belongs to routine. Here the MSN results are shown, different signals belonging to bases differing from each other are shown here. And the paper is named UHPLC, ultra high performance liquid chromatography, electrospray ionization, mass spectral analysis of purified flavonoids fraction from stem of Dendrobium denyayo, PAXT, and its preliminary study in inducing apoptosis of HepG2. So the extract obtained from this organism hold this ions. I mean, if it's flat, I'm not sure. Well, dendrobium. Okay, these three. Maybe from the leaves of this tree, the extract was obtained. Those extract was found to hold high level of flavonoids. Among those flavonoids, these compounds were found to exist, and that extract was claimed to induce apoptosis in HepG2 cancer cells. I think these are cancer cells. So based on this kind of approaches, it is possible to identify different components in extracts. So then the effects of those extractors on different cancer cells could be studied. Okay, next one. This is another interesting paper. In this paper here, we can see the LC-MSMS-based quantification method of polyphenols for valorization of ancient apple cultivars from Silento, whatever. Okay. Here, the color intensity is very close to each other. I hope you can differ them from each other. Different, okay, different varieties of apple were taken in this study, and their peels and fruits were studied, pulp, yeah, studied and compared. The point is, here we can see that the main component is in both peels and pulps belong to catechin and its derivatives. Also here we can see high level of phenolic acids. Here the dihydrochalcones ratio is very little, somewhere here and here only, here very little. And here we can see flavonoids and anthocyanins concentration also not very high. And the comparison can allow us to see that in the peel and pulp, here we can see the main differences belongs to catechins, phenolic acids, catechins and phenolic acid. Here we can see the catechins in the pills are much more compared to the concentration of pulps in Royal Gala and Golden Delicious varieties. So here we can make a conclusion that by using LC-MSMS tools, we can develop this kind of approaches and quantify not only some compounds, but also the class of different compounds. And here we can see protein digestion overview. The figure was taken from one ACS paper, American Chemical Society, In the first line, we are showing top-down proteomics. In this approach, we can see that proteins in mixture can be first separated, and then without any kind of digestion by using some enzymes, they can be subjected to mass spectral analysis. Here we can see mass spectral analysis, and further MS mass spectral analysis can be carried out, and MS results could be obtained in order to have more accurate results. So the first approach differs from the second one, middle down proteomics, by the inclusion of digestion by using some enzymes. Here, the authors can use different enzymes to divide the proteins into smaller pieces, so then they could be studied mass spectrometrically. And the point is here, when we use top-down proteomics without enzymes, the mass spectrometers has to have the ability to see the mass, have to see the molecular masses lower than 50 kilodalton, but higher than 20 kilodalton, for example. Yeah, by that, okay, based on this kind of capacities of the mass spectrometers, we can develop our own approaches. So in middle down proteomics, the enzymes are used to digest, and the molecular mass different from 2000 to 20,000 daltons can be studied, and both by using LCMS and LCMSMS approaches. And in bottom-up proteomics, the similar approaches as in middle-down proteomics are used, but the molecular masses of the peptides can be between 500 to 3,000 daltons. So by this 2,000 to 20,000 and 500 to 3,000 dalton range, middle-down proteomics and bottom-down proteomics differ from each other. Okay, and in this work, the authors were able to find the differences between the molecular masses of two fragments, and they made a conclusion that tyrosine holds a phosphate group in this position, because they found that between this molecular mass and this molecular mass, these two peaks might differ from each other by the phosphate group. Okay, the difference of phosphate group is not about that 40, and there are some other approaches. What the authors want to say that the A1, as shown here, the A1, okay, mass spectra of the precursor ions with 999 plus 2 and 1039 plus 2. This correspond to unphosphorylated peptide, this one, and its phosphorylated counterpart, this part. Okay, this belongs to unphosphorylated, and this belongs to phosphorylated segments of this peptide. So by using mass spectral analysis, it is even possible to identify the location of phosphorylation in different proteins, and this kind of results make us know about the locations of phosphorylations and understand the biological processes deeper. So the C here represents cysteine modified by alkyl corresponds to phosphorylate tyrosine residue, while the top spectrum contains only the unphosphorylated peptide. The bottom spectrum contains precursor ions that correspond to both unphosphorylated and phosphorylated peptides. The ratio of the two precursor ions indicates the percentage of phosphorylation of the peptide as well. So as we read more scientific papers, we can see uh, how effectively we can use mass spectrometry in research, in biomedical research, in pharmaceutical research, and other fields of research, including environmental sciences. So these are all for today. I express my thanks to you for watching this video, and I ask those who watch us on YouTube, please support us with your likes so that the video can reach a wider audience. And I express my thanks. Have a good day. Bye bye.